All right. Thank you, everybody, for coming back. Hopefully, you got a snack and a drink. I'd like to introduce our next speaker, Dr. Stephen Larson. So, Stephen Larson is the co founder and CEO of Medicell and is an expert in the intersection between computer technology and biological systems and is interested in how computer systems can help us understand fundamental principles of our life. Dr. Larson also serves as the executive director for the Open Worm Foundation. He served as the chief information officer of One Mind for Research, a nonprofit dedicated to eradicating brain disease by transforming the healthcare research system through public-private partnerships. He has worked as a professional software engineer for a major New York investment bank, co-developed a patent, presented at more than 24 forums, published in academic journals such as Frontiers in Neuroscience and Nature, and has had his work featured in the New York Times, The Atlantic, Wired, and MSNBC.com. His diverse educational background includes Bachelor of Science and a Master's of Engineering from MIT in Computer Science, as well as a PhD in Neuroscience from the University of California, San Diego. Thank you, Dr. Larson. Take it away.
Thank you very much, Samantha. Thank you to all of you as we're going into hour 2 of this workshop. I'm going to be presenting to you today some of the things that I've learned over my career about this space of AI and life sciences. As I do that, I also have a ringer. You get a 2 for the price of 1 here in the form of my colleague Phil. Say hi, Phil. Hi. Come off mute.
Hey everyone, nice to meet you.
Okay, cool. So Phil is with us here as well. Phil lives in New York and wasn't able to travel to speak today, but he will be giving the last part of the talk. And then when we go into our workshop, we'll have an interactive session that will also involve Phil. But Phil is our data scientist, one of our data scientists extraordinaire. He got his PhD in neuroscience from Mount Sinai, and he's continuing on to work with us and do postdoctoral work. So thank you, Phil, and I'm going to make you disappear. So we're going to talk about AI and data science in life sciences. So thanks to Dr. Malecki for setting up the context of AI and some definitions. And one of the diagrams that he had there was sort of like AI and machine learning and deep learning. Remember that one? Well, for the purposes of my talk, I'm also going to be talking about data science, which I would say is one circle even bigger than the AI circle from the way that I'm thinking about it, which is data science would also encompass AI, but then there's other methods that are data science methods that are not necessarily AI methods, although they largely will overlap. So that's where we're going to be speaking today. Just have this perfectly set up, but we will probably have to. Yeah. Okay. So we're going to start off talking about some AI trends and what's hard. Then I'm going to add to the really great definitions that Dr. Malecki started with a few more valuable concepts, specifically in Gen AI, that are very de jure right now. Then I'll talk a little bit about what we do at Medicell, working in digital life sciences, talk about data science challenges in life sciences that we've come across. Then we're going to get into a little bit of novel life science data collection methods that came out of some of the chats that I had earlier today with Dr. Kobo. And then Phil's going to come in at the end here and give us some data science examples that come from neuroscience, which hopefully pulls a lot of this together. And then once I'm done speaking here, Phil and I are speaking, we're going to kind of run a workshop in real time. And what we're going to do is we're going to ask the folks that stick around to actually talk about the data science challenges that you're all running into. So we're going to want to hear about that. Now, this is the largest group that I've gotten a chance to do that with. So we may filter that into like filling out a form we may pick. So I can't guarantee we get 100% of people, but we'll do our best. Okay. I think that right now, a lot of data science and AI is not, believe it or not, as much of a science as it is an art because everybody's doing their own thing. So I think just talking about it in a group, realizing that you're not alone, realizing a lot of people are having the same challenges is probably the first step. Sound good? Okay. All right, so some AI trends, things to consider when embarking on AI projects. All right. So this is the tough news. Okay. So research shows that most machine learning and AI initiatives are set up for failure. Darn. So this is where we talk about the bad news before we talk. So 72% of AI projects struggle with gathering sufficient high quality data, a critical barrier that often leads to project failure. This is an observation kind of in academia and in industry, but since we're talking about AI here, it's important to sort of say that, like, even though it's very buzzy to say, oh, let's go use a deep learning algorithm, the problems usually start with data. That's really where we ended before. What is high quality data? What does it mean? So that's important to think about. Also, 65% of organizations report a lack of necessary AI and data science expertise. So this is not well distributed skills. 60% of AI researchers encounter significant collaboration challenges when integrating AI into existing workflows. Significant collaboration challenges, right? And I think, a lot of that comes from the fact that these fields are still pretty separate. It's hard to be good at both things at the same time. Hard to be good at biology at the same time you're good at AI. And so both sides struggle to find the right sort of either folks or just teams or build teams that can kind of speak both languages. And then finally, and this is kind of more industry, but only 15% of AI projects make it from prototype to production, often due to siloed efforts within organizations. And yeah, so AI problems, which today we talk about it like they're AI problems. From my perspective, having been in this field and industry, depending on where you start counting, about 20 years, they kind of come back to challenges of data collection, data aggregation, data quality that have been with us even before the latest generation of AI algorithms ever existed. So it's just kind of important to like keep that together. It's not like AI came along and these problems suddenly emerged. It's more like assembling multimodal data sets is challenging, and applying any mathematical quantitative technologies to those are challenging. Okay, but there is some good news. Okay, so AI and data science is still going to drive the future. Okay, so using drug discovery as a proxy for kind of all of life science, which is a very poor proxy, but let's just because we were pulling some data. So it's expected to accelerate generative AI, and that's the smallest part of the AI field is expected to accelerate drug discovery. 10 to 15% reduce cost of drug development and 25 to 30% increase R&D productivity. This is what a consulting firm says. I think that, depending on where you're counting, it could do a lot more than that. And then sort of in parallel to that, not exactly about AI, but in terms of the ways folks are thinking about data, institutions that are using cloud platforms are seeing 50% improvement in data accessibility, 40% increase in data processing speed. And by cloud platforms, I sort of mean places that are in between your individual computer, maybe the computer that's collecting data in your lab. Cloud platforms that are places where data can come together and places that you can come back to, collaborations can be formed around. This is something that I'll talk to you a lot more about in detail. It's sort of something that we've been working on for quite a while, even before this latest excitement in AI algorithms. Okay, so valuable concepts in AI that can be applied to the life sciences. Okay, picking right up where Dr. Malecki left off, I want to talk to you about 3 more ideas in AI that may be helpful to you going forward. The first is foundation models. Okay, so foundation models are large-scale learning models that are trained on a lot of different data, and they can serve many different downstream applications, offering general purpose capabilities. So at the core of things like ChatGPT is a foundation model, okay? And pictures like this sort of help to give you a sense of that. So going into things like foundation models are text and images and speech and structured data and 3D signals. And after a lot of training, and in the kind of foundation models that we're most familiar with, like ChatGPT, we're talking about like millions and millions of dollars worth of training, millions and millions of dollars of compute. What comes out then is a thing that is very flexible and can do many different tasks like question answering, sentiment analysis, information extraction, image captioning, object recognition, instruction following. It's a little bit the holy grail. And it's, for those of us that have been studying AI for a while, honestly, it's like sounds like science fiction. It's kind of science fiction. But at the end of the day, it is a neural network model, like the picture that Dr. Lucky had up here earlier. So it's essentially just layers of nodes, very big layers of nodes. But the magic is that with internet scale data and cloud level compute, we are able to put these two things together to make these very, very impressive neural networks that are then able to do lots of general things. So foundation models, there are many different kinds of foundation models. One behind ChatGPT is more general reasoning for language. But as I'll get to a little bit later, you can also start to think about, well, what would a foundation model in biology be? What would a foundation model in the domain of biology that perhaps you're interested in or that your lab works on? Like, what would they look like? So I'll leave you with that thought for a moment and go on to the next idea. Transformers. Okay, so transformers are a type of deep learning model architecture primarily used in natural language processing and sequence modeling tasks. This was introduced by a paper called Attention is All You Need by Vaswani et al. In 2017. And that paper is credited with kind of kicking off this latest revolution in AI. So what is a transformer? Just real briefly. Okay, so it's at the highest level, think of it as the middle block in a block diagram where you give it some input. It is, again, essentially just a neural network, and what comes out is some output. In this case, this transformer can take in French and spit out English. Okay, if we unpack that box one more level, a transformer is made up. So now this blue box here is the same blue box here. Right? So it's a combination of, on the one hand, a set of encoders, and on the other hand, a set of decoders, okay, that have a relationship to each other. So it's just one step more complicated. So first it's just a magic box. Then it's a magic box with two other magic boxes inside of it. One's called encoders, the other called decoders. And then if you unpack that one more level, you find out that actually what a transformer is, it's a series of encoders that are related to a series of decoders, okay, that then give you your output. And so within those boxes, then, if you peel open what is an encoder and what is a decoder, the transformer is composed of a layer that does self-attention, which is that unsupervised learning thing that we heard about earlier, and then a feed-forward layer, essentially a supervised learning thing, which we also heard about. So those two things go together. And the decoder combines those two things, the feed-forward layer and the self-attention layer, with this encoder, decoder, and attention layer. I'm not going to go in a ton more detail of transformers, but if you'd like, this thing here called the illustrated transformer is a very nice, accessible way of starting to get your head wrapped around what transformers are. Okay, so, and the third portable concept that I want you guys to walk away from in terms of expanding your AI vocabulary are latent representations. And so that is compressed or transformed versions of input data, capturing its essential features in a lower dimensional space, often learned through models like encoders, curational auto encoders, or during the training of other neural networks. And so what is a latent representation? So essentially, this more refers to kind of the content of what transformers are storing. So it's a different word. It's not a different thing. It is a lens on what you are getting. So the latent representation, it's like the weights that are in the network that you learn in a particular domain. So you might train up a transformer that you're giving it inputs of some sort of biological data, and you're getting out some other outputs of other biological data that then is transformed with those encoders and decoders that we just talked about in terms of transformers. And then you might be able to do things like cluster, but also predict drug responses, or also predict a perturbation in a life science sense. And this reference here, How It All 2023, is where this diagram comes from, and it's pretty nice also talking about how, some biological applications of this. So then I just want to tie these together. Okay, so these three things are kind of related, right? So let me just read this out loud. Foundation models leverage transformers for their architecture and often produce latent representations as part of their processing. Okay, look at that again. Foundation models leverage transformers box diagram that you're talking about for their architecture, and often produce latent representations as part of their process. So if you understand that the latent representation is like the knowledge, even though you don't really look at it because it's just a series of weights and numbers. What it is that it can be a very useful thing for other steps of reasoning or doing very useful things for you. So let's just kind of apply these for a second, okay? So that you kind of get a little bit more feel for what I'm talking about. Okay, so foundation models that you may be familiar with, and again, this is kind of underneath the hood, but things like ChatGPT, Claude, Llama, okay? For language, those are the foundation models. Under the hood, right, the architecture that's used, okay, is going to stay the same independently of which language model you're talking about. You're basically using the same kind of transformers in all of these cases when it's regardless of which language model it is. The difference will be the data and the learning rules. So even though all three of these things kind of do the same stuff, they're all trained a little bit differently. The data sets are a little bit different. They have a little bit different output quality, that kind of thing. And the latent representation is sort of encoding the semantic and syntactic information of the text into dense vector spaces. So we talk about the latent representation of ChatGPT as this, it's just like it knows language and it knows facts and it knows things, but they're encoded in the weights of the transform. Okay? All right. Now let's go to image. So I don't know how many of you have generated images using AI recently. ChatGPT does that now too. But things like DALL-E and Midjourney. Okay, so they're also foundation models for those. You were trained on a bunch of data. They spit out new data. Generative AI, amazing. Now, also the transformers, same thing, right? Like the fundamental architecture kind of stays the same regardless of which one you have. And the transformer architecture is not so different from what you're getting even in the language models. Okay, so very different kinds of outputs, very similar underlying technology. But the latent representation, if we're talking about images, would be what we're saying we're encoding there is this visual information or the ability to transform from a textual description, I want to see a penguin riding an iguana, to pixels, right, on the screen. So it's kind of encoding this text to image thing, okay? Again, kind of the best we can do, but we'd say that's the latent representation that you get in these image things. So what are they for biology? Well, there aren't really many of them. There's starting to be some, okay, in the spaces of genomics, proteomics, but it's very, very early days. And we're talking about things that people are sort of putting out on GitHub repos, but things haven't quite reached the level of mass use. But the reason that I wanted you guys to be thinking about all three of these things is that there's nothing that stops us applying biological data into the same space and harnessing and taking advantage of generative AI in this way. So again, the transformer architecture under the hood can be quite similar to what we're using in these other cases. And then what gets really interesting is to think about, well, what are the latent representations that we could build if we're going about the work and building foundation models for biology? Could be capturing essential features of biological data like gene sequences, protein structures, and cellular states. And so at my company, Medicell, we're working with organizations that are pushing on things like reimagining virtual cells in the context of foundation models, as opposed to some of the computational biology methods that I grew up with that are more about systems of differential equations and that kind of stuff. So just interesting to think through these things, and we can talk more about how to apply these things in the future. Okay. So now I'm going to turn to a little bit of working in digital life sciences and just briefly talk about what we've done at Medicell before getting into the data sciences. So I've had the great honor coming out of a PhD in neuroscience to have built a company with 3 people with 0 investment and just like, I don't know, to get to work with all these kinds of organizations. And I guess what I like to mention about this is it's very cross-sector. So, at the same time that we work with major universities that are on this list, we've also had the chance to work in preclinical research with major pharma companies. We've worked with some pretty prominent nonprofits. We've worked with some medical research hospitals. And the thing that ties it all together is essentially helping scientists do their work better. Okay, like that's really what drives us. It drives me. A lot of the work that we do is building custom code, work for hire. We work a lot in the open source. We're not a software company that, builds a product and then sells you the code. We tend to just offer our expertise. That's really what we do, and that's what's been fun. As a result of that, we've worked with many, many different types of data. We did start out life as a neuroscience sort of related organization, and we tended to take a lot of neuroscience projects. But as time has gone on, we've broadened out to more general omics applications. So a lot of different kinds of data we're very comfortable with and familiar with. And we like to put together our smartest sort of PhDs on the life science side with some of our smartest AI and AI PhDs and software engineering master's folks. So what kind of stuff have we built? One of our earliest and also longest running projects is the virtual flybrain that we have worked together in a collaboration with originally EMBL and now University of Edinburgh. And this is an example that's very visual, which I like to show, of connecting and combining different modalities of data into a single software usable interface. So this is a cloud-based application, so you can go to it on the web, Virtual Flybrain. In this particular piece of software, we have organized everything around the 3D atlas of the Flybrain, much like Google Earth. It's like a 3D atlas of the planet. So, and just like Google Earth, it has multiple layers of information. On top of it, it's got information about roads, it's got information about elevation, it's got information about I think the weather, and it can all be overlaid on the same coordinate system. Similarly here, we've laid out information about the Flybrain all coordinated together in with spatial coordinates. But in addition to that, we also have layered in knowledge about what we're looking at, whether it be what part of Flybrain we're looking at, or what the name of a neuron of interest is, or what what the genetic profile of a particular stain in this data set is showing you. So what sort of proteins may be localized to a given color that you're seeing here.
So what kind of data you get like an input to generate these 3D models?
Yeah, so there's 2D slices taken from like confocal imaging, and is one of the main ones. There's immunohistochemistry, also like slices through brain, and then I believe we also have individual cell morphologies that were taken, so a lot of microscopy data, and then connected with some genomics data sets as well.
What species of fly is it?
Drosophila, Melanogaster. That's right. Cool. Okay. And this is a similar idea for human brain that we did in partnership with a pharma company. So for preclinical drug discovery, this pharma company wanted to, was very interested in combining 3D and 2D imagery. And we love making interfaces that are, zoomy and flashy. And not just for those reasons, but also because I think that there is a quality of putting things in 3D that you actually get more insight, because I think our spatial reasoning, once we see things in 3D, is going to be better than just looking at things in tables and charts, perhaps, right? So this is a very similar idea where we're also connecting things from the Allen Human Atlas with knowledge of anatomy, and the part of the Allen Human Atlas is also some gene profiles that are into this. So these are fun pictures to show. These are things that probably don't apply to things that you guys are going to be building here, but I just want to give you that as a sort of a reference point for what we've had a chance to work on. Okay. So now let's go to more practical data science challenges in life sciences. So what I just showed you is kind of been the history of our company, where we started out building these larger scale software projects. But in the last, say, 2 or 3 years, we really saw the need to address these practical data science challenges that aren't coming from folks that have already collected data and want to assemble them, but folks that are like just collecting data and want to analyze it. So really working with experimentists. And so what we have hit on is, this sort of research pain point, which I think may be familiar to some of you in this room, which is we need an analysis completed and you don't have the expertise in the lab to do it. What do you do, right? We found this just from talking to folks over and over and over again, hearing this problem. How can labs get high quality data science support? So off to one, bring in students, postdocs, collaborators to perform the analysis. Okay, so but we also hear challenges of that repeatedly. So a few of them are that this can be time consuming, and that another one is that the specific analysis may not be the sole focus. You want to analyze a thing right now, but you set up a whole collaboration to get that one thing analyzed. That might not, that may be great, and I participated in many of those collaborations as a graduate student, so no problem with that. But might not, but maybe there's a more efficient way. And so what we started to think about was, could you engage remote data scientists to work on particular data analysis problems as needed, with the idea that the remote data scientist would focus on a particular analysis problem and literally do it for only as many hours as was needed to solve it, and then go home. Okay. So we started to think about how that was combining with some of these cloud technologies that we were making. And instead of having flashy 3D visuals, we actually stripped back the technology, made it much simpler, essentially thinking about what could you do where you basically just have very familiar open source software like a Jupyter Notebook, right? Combine it together with the ability to log into it, add some storage space, maybe throw a Linux desktop onto it, and then just use that, because we were very good at spinning up virtual machines in the cloud. Just use that as a rallying point, a hub, if you will, to have a conversation with a scientist and say, hey, we can bring you a data scientist. If you bring your data to the party, we can combine those things together in an analysis environment that you can log into, and we can log into, you can have a data scientist log into, and we can help you do your data science in this third space, in this cloud space, this online space. We found that this was weirdly important because when it's your personal computer, right, there's always going to be problems. Oh, the folders are somewhere else. It's, the data is, I updated the data. Now the analysis is broken. Things like this, right? Whereas when we had a shared space where literally data scientists log into the same machine with everything is the same as the same machine that scientists are logging into, then a lot of those things could be solved. Okay, and not have those symptoms. So we built a way to start to do this. And so we call it Cloud Workspace as a platform to deliver remote data science help to scientists. And so these are the kinds of things that I'm saying. So we can connect cloud data. One of the things that folks like is the ability to say, if you have your data on Dropbox or OneDrive or Box.com, right? You can sync it to a workspace, and only those parts that you want to share can be synced. And that helps even if the data are moderately large. We connected this personalized virtual desktop, so you can run additional apps. You can run MATLAB on it if you want. Then we could help folks build their analysis pipelines in JupyterLab. We could, tackle these specific data science problems as needed through online video and chat support. And also, hopefully, we were finding that labs really liked it because even within the lab, multiple lab members could log into that same workspace and share. And that also started to reassure PIs that when their students graduated and left, that they wouldn't have to go rummaging through their laptops to go find their data sets, right? It would still be there in some amount of longevity and reproducibility. And we also, started to find that it could work for both small and large data sizes. For small and medium data sizes, you can log into it, from your laptop or desktop computer. And for larger data sizes, we've worked out ways that makes it easy to create high bandwidth connections, maybe even directly from the network file system that is connected to your experimental rig to directly sync. And then the workspace can be pulling from that rather than having to necessarily even upload 100% of it. Okay, so that's all. These are all very practical process things, but I'm gonna now, so this is okay, it's the method of how you do it, a bit the social process of how it works. So now I'm going to talk a little bit about getting into the data and kind of closing with some specific data science examples of what we've done when we've collaborated with folks in this way. And then Phil's going to talk about that. Before I do that, though, I want to address some novel life science data collection methods that we are starting to see. We have had the opportunity to work with some organizations that are looking into the future of where AI and life science might go. And they are thinking a lot about data collection methods that can help to bridge gaps between different data sets. And so I would have the chance to talk to Dr. Kobo earlier today and his lab, and he started talking about like, I have all these different data sets, I don't know how to pull them together. And so part of it is using methods to, collect data in good ways and integrate them. But some of the things that we are starting to see here, and I'm going to only go over these very quickly, okay, are things like spatial omics approaches to even collecting data. Now, I say these things are still up and coming because some of the techniques that we're seeing are still like too expensive, right? Need to be reduced to product. Okay, these are like early days, but we're inspired by it and we're looking at it because we think that, in a future looking way, data that may come out of methods like these may be things that you build foundation models out of, maybe things that, we're better able to apply AI to. So in spatial omics, this is sort of an umbrella term that can, consist of many different things. I'll just sort of mention that there's, sample processing that might involve either doing flash frozen or FFP embeddings. You mount them onto slides. You would, do targeted data acquisition. Still using transcriptomic methods, right, and sequencing, as well as potentially mass spec. And then what we're seeing in these methods using, there's a variety of different pipelines, which is my next slide, but can start to include some deep learning-based options. This is not without challenges. This is not a panacea, but I would reference this paper here, Park et al. 22, if you want to get a good overview of some of these methods. Some of the data analysis that we're seeing here. So folks using reference atlases like HubMap, as well as some of the other ones that you can just read here off the slide. There are challenges. There's not real standardization happening yet, other than, we're seeing this OME TIFF as a base image file. Again, this Park paper will also help you see some of the challenges that are coming here. There is heavy use of UMAP and deep learning to interpret the high dimensionality, but that can also be dangerous. So, mind that. And then also there are very large data sizes that are coming out of these sort of spatial multi-omic techniques. The other sort of category of data collection that I wanted to talk about was optical pooled screening. Again, a bit of the even less developed than multi-omic, spatial multi-omic processes, but these will typically involve starting with a pooled genetic library, collecting phenotypes via imaging, doing sequencing of perturbations, and then finally doing phenotype to genotype correlations. The data analysis for this is again early stage, but there are some pipelines here we can talk more about in the future in collaboration. Some image acquisition, some phenotyping and screening, and there is some predictive modeling starting to happen. So I just wanted to kind of name check these honestly and not really go into a lot of depth, but I think the important thing here is that we are living in an era where like single cell transcriptomics, all the rage, RNA-seq, amazing, right? But folks that we are working with are starting to realize even just the limitations in data collection if you do want to make larger statements at the systems biology level, at the level of tissues, at the level of like, well, what position was that cell in and that kind of thing. So so we can have future conversations about which ones of these very nascent technologies might even be better for machine learning based approaches. Okay, so now I'm going to let Phil talk to you about some data science examples that we're seeing in neuroscience. So a lot of our data science work that we've been doing in the last 6 to 12 months has been drawn from neuroscience, in particular, a strength in calcium imaging. I know there were some folks that were interested in imaging earlier. So I'm going to drag Phil's window over here, where he has this pulled up. I'm going to full screen this. So Phil, you are now front and center for everybody, even though Phil cannot see any of you, but I promise it's a very good looking crowd. And all right, and I'll be quiet and let Phil present to you guys.
Awesome. Thank you, Stephen. So yeah, so I'm gonna quickly go over some example projects we have done within Medicell, and mainly focus on neuroscience. So there are mainly two ways we sort of operate in this regime. The first one is we perform concept analysis on behalf of scientists. So we, the scientists ask a question and we go in, we either analyze the neural or behavior data. The second mode of operation is we build pipelines so that the scientists can use on their own without, us being there anymore. So currently we have pipelines focusing on calcium imaging, photometry data, or behavior extraction. But we have more coming up, and that's kind of the other way we mainly operate. Here are some example projects we have done in the first regime, where we sort of perform the analysis following a question from the scientist. So we have collaborated with DGIST in Korea and investigate the neural activity during a reversal learning task. Specifically, we look at the reward signal in the brain and how they interact with animals' choice during the learning. We have also collaborated with McGill and investigated inhibitory control in the brain in another behavior task called go and no-go task. And specifically, we look at how the inhibitory signal correlate with animals' behavior when they are making the choice to withhold their impulse to go. We also collaborate with Max Planck Institute, Florida, and PFI, and we investigate internal state change during a feeding behavior of animals. So specifically, we look at how the population neural activity change when the animal is more hungry compared to when they are more thirsty. That's another interesting project. And lastly, as another example, we collaborate with another group, NPFI, and investigate dog-type specific response during social behavior. And for this one, I'm just gonna quickly jump into a little bit more detail on this project. So here is an overview of the project. Here, the scientists developed a novel technology to record calcium neural activity with a miniature microscope while the animals are freely behaving. And when the animals are behaving, they put them in a social context so they can perform some sort of task and have social interaction with other animals. And after that, they put the animal on a tabletop microscope, and they can image the same field of view. And that allowed them to look at specific cell type information, and they are allowed to image a lot of colors, I believe up to 10 colors in the same field of view. And that gives them information about where the cell is projecting, what kind of input it's receiving, what kind of output it is outputting. So that's the general technology that scientists developed. But as you can see, this is a complicated data set, right? We have the behavior data, we have the real-time neural activity, and we have the static label of cells. So the scientists really find it a little bit challenging to deal with this complicated data set. And this is where we jump in to help. Our task here in this project is to find distinct response of different type of neurons responding to different type of behaviors. So quickly go over some analysis we have performed in this project. We have classified the different types of behaviors. We have performed some deconvolution of the real-time neural activity, so the signals can be estimated more accurately. We have some clustering analysis, dimension reduction analysis, both supervised and unsupervised, trying to see whether cells from the same brain region tend to have similar activity that can be sort of clustered in the high-dimensional neural space. And here is a quick look of what we end up coming up with. This is the sort of the final project of this project that actually end up going to the paper. So we have this visualization, this plot here, where on the top we have different brain regions, and that information we obtain from the static labeling of cells. On the bottom row here, we have different types of behavior, where we classify behavior using the behavior video we collect during experiment. And these lines, these colorful lines connecting the top and bottom row, indicate what proportion of cells in that brain region are responsive to a specific type of behavior, right? That's why you see this, they have this very complicated graph almost, and they have different widths that indicate different proportions. So this plot really kind of highlights the capability of handling the multiple data modality, and it also highlights the strength of the technology. You can investigate neural activity in relation to behavior, but also have the information of what kind of neurons is performing this kind of response. So that's a quick look at the sort of one mode of operation, and then we have some pipeline that the neuroscientists can use to operate on their own, and they can use the pipeline to analyze their own data. We have calcium imaging analysis pipeline based on the Minion package.
Hey, Phil,
We have, yes.
1 second. We have a question. Yeah, go ahead.
Oh, sorry.
Yeah, very interesting. To the previous study in Florida.
So the previous study, yeah.
I can choose like the previous slide.
One more back.
Yeah.
That slide.
Yeah.
So here it's amazing. No, I mean, you have at least 4 different methods. And it come my question to about the quality of the data. So behavior is been scoring, so probably it's becoming more biased because at the end it's a human being looking at that behavior. And then you have much like calcium activities, a lot of data. So the 4th set of data are quite different. No, some are more parametric, some are more non-parametric. And then eventually any 4th set of data you are able to aggregate or depend on the set of data that you have. Or if I give you other 4 different set of data, you eventually will come with something. That, I don't know if it's clear my question.
I heard you doing a good job of explaining
Yes,
what you see.
Yeah, when I
The
see-
question part, I think I missed that though.
And then you have the power to analyze this data because this is this particular set of data.
Mhm.
That is calcium, and probably you are familiar with calcium, but cell, and probably you are familiar with the responses, or eventually any 4th set of data, and they have that high quality, you eventually can to put together in the way that you have put.
It's
So that's the question, whether how applicable is this approach to different set of data?
okay. Phil, did you hear that?
Yeah, I'm not sure whether you want to answer that because
Well, I mean, I'll give like at a cursory level, so obviously every study is different. And,
Yeah.
and we were talking earlier
Yeah.
this morning about how, there's not a one size fits all solution to data analysis.
Yeah.
That there's no getting away from, having people that deeply understand the actual experimental methodology that was used so that they understand the caveats of the data when they're doing that. At the same time, in my observation, there's also a skill set that I think Phil has just from having done many different kinds of studies of sort of starting to organize data in a way such that you can bring multiple modalities together, even when 1 or 2 of them might be something you hadn't done a lot of work with in the past. So that's my very generic answer. Maybe Phil can give a better, more specific one.
yeah, I think that's basically what I have in mind. The other thing I want to add is, yeah, it's true. Maybe, personally, I'm more familiar with neuroscience data, but we're kind of presenting more like a mode of operation. We have this cloud workspace where any data scientist, other than me, can jump in and help with any type of data. So, if we have other types of data that I'm not the expert, we have other people in the company that can jump in and basically operate in the same way. That's what we're presenting here.
So yeah, we have folks that are a bit more coming from the omics world, for example, into neuroscience that that we can also bring to bear. So yeah.
Amazing. Thank you.
Cool.
I like this one and I like much more the next slide. But the next slide is the consequence. This.
That's right. That's right. Cool.
Yeah.
Great. Yeah, no, great question.
Okay, cool. So, once we move on from the example here, we have different pipelines that the scientists can use on their own. I think we went over the calcium imaging and the photometry pipeline. We have a package called Simba that's also available on the workspace that can help with behavior extraction. Here are some quick look at, kind of how they look when you actually use them. All of the pipeline are based on Jupyter Lab Hub. It's an interactive interface where you can run the code in Python, and you can change the code parameter, and you can see the visualization and see the result all in the same place. We believe this makes everything more user-friendly to use. We also perform customization of the pipeline for scientists. So here we actually collaborate with a pharma company called Maze, where they perform very high throughput calcium imaging, a lot of wells, and they really need an overview of the extracted neural footprints across all the wells at the same time. So we build this visualization for them so that they can look at all these parts at the same time. You couldn't see here, but really it's interactive. So when you zoom in on cells, they zoom in on all the plot, so they can use this tool to analyze their data and visualize their data better. We now have a photometry pipeline. Again, that's also based on Jupyter interface, and the user can interactively see where the events are happening along time, and they can see the neural activity that's time locked at different timestamp in the behavior. And we're actually collaborating with Dr. App's lab here at UCalgary to build customer support for behavior timestamps. Lastly, here's a quick look at the Simba pipeline, where you can use this to label the behavior and classify them into different types of behavior. For example, different social behavior are shown here when animals are either attacking or interacting with other animals. And we have this interactive view where after running the pipeline, you can see the result of the tracking. As a summary, we have remote data science as a service, and we believe this can really facilitate and help scientists with their questions, either by performing analysis on behalf of the scientists or by building pipelines that can be easily used by scientists in all backgrounds. And with that, I would like to hand it over back to Stephen for some closing remarks.
Great. Thank you.
Thank you.
Moving you back over. Those are the data science examples. Okay, so yeah, I think we've sort of we've gone a long distance now from talking about challenges in AI. We talked a little bit about some key concepts in AI that may be useful to you in the future: foundation models, transformers, latent representations, as we start to think about how they can apply into biology. I'll talk a little bit about some of the ways that we've brought multimodal data together in large software. I touched briefly on the advanced data collection methods that might be helpful in the future for assembling data about systems together that may be more applicable to veterinary science in the future. And yeah, so we as a company traditionally make software to present, collaborate around multimodal data neuroscience, but we've gotten closer to the analysis of the data. We're helping labs around the world get data science help that they need and also work with AI. And examples of work that we've done have included calcium imaging, photometry, behavioral analysis, and I haven't mentioned here also a lot of omics projects too. So with that, I will say thank you. Now it's currently 3:15. We have till 4 p.m. So what I would suggest is that we take a 5-minute stretch, and this QR code, rather than being the code to like to email me or something, it's kind of will email me. This is where anybody who wants to stick around is invited to briefly write a couple sentences on any data science or AI challenges that you are having. It's like links to a Google form. Okay, so you go to this, you get a Google form, and I will bring up the Google form right now, right here in real time, and I will go through them, and we will talk through them, and we will just talk until 4 p.m. when folks have to leave. And I will do this also with Phil, and we will, as I mentioned at the beginning, just see what thoughts emerge, what thoughts or ideas that we have about how to get over the data science challenges. So I will right now just invite anyone who wants to do that to just do that and stick around. We'll start again at 3:20, and if this is your ending point, I will say thank you very much.